Hello, my name is Lisette Wa. Thank you for joining me from this presentation, which is being recorded from my yard in Moscow, Idaho. So I'm a professor at the University of Idaho, and my co-author is Nico Balkenhol. So I want to start by giving you a background on wolverines. So wolverines have a circumpolar distribution shown here, and they're found in a narrow climatic niche, which depends on snow. And they are the largest member of the family Mustelidae, and they're highly mobile. They standardly disperse hundreds of kilometers, and males have been known to disperse over 800 kilometers. But they are found at low densities across their range and are a species of conservation concern. So focusing in on North America, you can see their current range here in dark green and their historic range in light green. So they have had a decrease in their range due to habitat loss and overharvest, and also probably due to changes in climate. And in the lower 48 states, there are only small populations of less than 300 individuals remaining, and they've been petitioned for listing under the Endangered Species Act. However, in Canada and Alaska, they are still harvested for their fur. There have been genetic studies of wolverines in the past, and these studies have found in Alaska, they're basically panmictic with no structure, high gene flow, and high diversity. But as you move south, genetic structure increases and genetic diversity decreases. People have also detected the impacts of the Trans-Canada Highway in terms of limiting dispersal and more for females than males. And we have observed highly structured populations in Montana, Idaho, and Wyoming. So the results of some early work done by my master's student, Chris Zagelski, showed that here in Idaho, Montana, and Wyoming, we detected 3 different populations on what is a small spatial scale for a wolverine. And you can also see that there are migrants between populations, like these individuals in yellow here would have been born and migrated to this region. Now, after doing this research, we were very interested in determining why there was less connectivity in the lower 48 states. Could it be due to decreased snow cover, topographic differences, or anthropogenic influences? To address this, we generated a larger data set by working together with others who had sampled wolverines, these 5 individuals shown here, and we were able to generate a data set with 210 individuals and 16 microsatellite loci. And the first thing we did was test the hypothesis that snow and snow depth coverage was an important predictor of connectivity in wolverines. And we published that work in 2009 in Ecology, and there's other ecological work showing the importance of snow for wolverines. This particular research that I'm going to talk about today is now available and online early in the Journal of Mammalogy, and we wanted to look at a larger suite of environmental variables and ask what is their relative importance in shaping genetic structure, and also evaluate whether scale effects were detectable and did they change our answers to question 1. So first, we calculated pairwise genetic distances between individuals, and we used the Miller's D genetic distance, and then we also needed to calculate effective distances. So first, we developed a set of variables that we felt would be potentially important for wolverine dispersal, shown here, and then developed hypotheses about how these would influence dispersal. For example, we expected increased snow depth to increase dispersal for wolverines and genetic connectivity. Also, we expected conifer forest cover, the existence of forest edge and ruggedness to increase dispersal, and anthropogenic variables to decrease dispersal. And we expected elevation to be more complex because their wolverines are thought to have optimal dispersal right at the tree line. So we also needed to evaluate the functional responses of habitat suitability and landscape resistance, which could be linear, or this higher exponential, or the lower exponential, or call this also the upper exponential. And we modeled this in a least cost distance form and also in an isolation by resistance form. And we tested all these possibilities using Mantel test, and then found the most suitable effective distance and conversion, which in most cases for effective distance was isolation by resistance, and in most cases the conversion was higher exponential. And then we also included a seventh variable of geographic distance or space to account for distance. So in terms of our question of the relative importance of various environmental variables in shaping genetic structure, we used hierarchical partitioning based on multiple regression of distance matrices. And then to evaluate scale effects, we used multivariate correlograms. So first, I will show our results for looking at the relative importance of various environmental variables. First, I want to highlight that space had a 10% contribution to explaining dispersal habitat suitability. And however, snow had the strongest effect. Over 30% of the contribution was due to snow, followed by terrain ruggedness, housing density, tree line, road density, and edge. And so we can see these four variables had a more important influence than geographic distance. In terms of looking at scale effects, we used a spatial autocorrelogram looking at pairwise relatedness between individuals over different distance classes, with the smallest distance class here, the largest distance class out here at 15. And what we found is that we have positive spatial autocorrelation up to a certain distance class, and then negative spatial autocorrelation here. So we divided our data set into different scales. We're going to define a small scale as being up to 230 kilometers, where we have positive spatial autocorrelation, a large scale beyond 420 kilometers, where we had negative spatial autocorrelation, and rerun our multiple regression on distance matrices analyses. And the results from that are in the next slide, shown here. And so again, geographic distance is indicated by a line, and its effect is shown here. And interestingly, we see at the small spatial scale, similar to what we found with all data combined, snow is the most important variable, followed by ruggedness and housing density. But when we evaluated at the large scale, housing density became to have the strongest variable contribution, and followed by ruggedness, for example. And then road density had a similar contribution as snow at this spatial scale. And these full models explained 13.5 to 14.7% of the variation in genetic distances, which is a good level of explanation for an individual-based landscape genetic model. So moving into our results, or conclusions, we combined all this together to develop a permeability model that you can see here. And our overall conclusions is that the finding of Swartz et al. Of snow being an important variable was corroborated. But when you look at larger spatial scales, housing density was a very important variable, and accounting for scale effects increases the biological meaningfulness as well as the conservation relevance of our landscape genetic analyses. For wolverines, we found that climatic, topographic, and anthropogenic variables were all important in explaining landscape genetic relationships, and that the importance of variables, the relative importance, varied by scale. In terms of thinking about the conservation implications, climate change is predicted to decrease snowpack in this area, and development is predicted to decrease connectivity in this area, which does lead to questions about long-term persistence of wolverines in the lower 48 states. And with that, I'd like to acknowledge different funding sources, and thank you for your attention, and I'll be ready to answer any questions.